Okay, so we're talking our way through the immune system, and we covered the first line of defense, which are your barriers, the skin, mucous membranes, secretions, and your microbiome. And then if something gets inside, we have internal defenses. We have our first response to infection and injury is inflammation. We have our natural killer cells, which are primed and ready to recognize our corrupt cells, so virally infected or tumor cells. We have a complement system that complements phagocytosis, inflammation. It has a membrane attack complex. And then most things are going to be removed by our phagocytic cells. Neutrophils are the first ones to get there. You see them in an acute infection. And macrophages are slower. They're longer lived, so you'll see those in chronic infections. So today is all about the adaptive immune response, which is going to be very targeted, very specific, and the branch that actually gives us immunity to disease. All right, so let's have an introduction.
The adaptive immune system provides a more targeted defense. Dendritic cells are like intelligence specialists. They collect pieces of the invader at the site of the battle. Then they travel to the nearest lymph node, triggering a cascade of events. Inside the lymph node, the dendritic cell presents pieces of the invader to T cells, the immune system's communication specialists. In turn, T cells activate B cells, training them to form antibodies, the molecular weapons that target the invader specifically. The immune system now mounts its ultimate defense. B cells release millions of antibodies, each antibody's shape tailored to identify and neutralize the enemy. The immune system deploys these tailored weapons until the infection is wiped out, ready to respond even faster the next time.
Okay, so this is the most effective branch of the immune system. This is the branch that is taken out if somebody has AIDS, so acquired immunodeficiency syndrome. It is carried out exclusively by lymphocytes, although there's going to be a bridge between our innate immune system and our adaptive immune response, and all the white blood cells, they do talk amongst each other. It is going to be targeted, so one lymphocyte will respond to one particular antigen. Lymphocyte, they do chicken, they do it right, they just do their one thing. So each one of them will recognize something, but you have a whole army of them that are diverse. It is called adaptive because each time you are exposed to something, either through vaccination or disease, you will leave behind memory lymphocytes, which are very long-lived, and so they'll be available next time you are exposed to that same infectious agent. And a good immune system, when it's functioning properly, can tell the difference between self and non-self. And so that's called tolerance. When you have autoimmune disease, that is a lack of self-tolerance. And so that will be at the end of our day. Yes?
Just a question about vaccinations. Why do people sometimes get sick after they get vaccinated?
We're going to talk about vaccination. Can I cover it when we get there? Okay. This is a good slide. This is a money slide. Okay, so one of those, if you get this, I'm going to ask some questions about this. All right, so this picture, so our adaptive immune response, there's two branches to it. One is called cell-mediated immunity, the other is humoral immunity. So cell-mediated immunity is going to be carried out by T lymphocytes. T lymphocytes made in the bone marrow go to the thymus. We discussed the functions of the thymus. The T lymphocytes, they are going to recognize our cells that have been infected. So either virally infected, there's a bacterium living inside them, or they've mutated and they are now becoming cancerous. And so the cell-mediated immunity is effective against antigens within cells. I'm also going to introduce to you antigen-presenting cells that are required for this branch of the immune system to function. The two big players in the cell-mediated immunity are the cytotoxic T lymphocytes, and their name is kind of telling you got cell toxic. These are going to be assassins. These are going to be killers, and they will recognize one thing, because they're specific. If they find their one thing that that cell is infected with, then they will kill that cell by cell-to-cell contact. So there's a whole lot of cells in this side: cell-mediated, cell-to-cell contact, cytotoxic. The other population are called T helper cells, or T lymphocytes. This is the general, the coordinator, the conductor, whatever picture you want to paint of your immune system. They do a lot of talking, but they are necessary for both to have activated cell killers, so recognizing our virally infected or cancerous cells, and notice these little arrows. They're also required for the other branch of our adaptive immune response, which is called humoral immunity. Humors is that old-timey word for all your liquid parts. Antibodies are soluble. They're immune proteins, so immunogens, and so that's how it got its name. Antibodies are made by the B lymphocytes, and antibodies stick to the outside of things. So this humoral branch is very effective against targeting things outside of our cell, so antigens outside. B cells, these B lymphocytes, do not require this presentation that T cells do. B cells can run into their antigen first. Okay, so they don't need to be presented. And we're going to talk about antigen presentation in just a moment, but that's a key difference between cell-mediated immunity and T lymphocytes and B cells and humoral immunity. When a B lymphocyte meets its antigen that it's targeted to fight, it's going to get activated, and we will get a clone army of plasma cells. Plasma cells make antibodies, and antibodies are going to stick to things specifically. And once something is covered with antibodies, it's done. It's going to be neutralized, it won't be able to hurt us, and it will be targeted for destruction. So that's where we're headed. But this is like, do you understand this picture? If you can explain it to somebody else, then you're doing well. All right, so what makes a good antigen? Like I said, there are things that the immune system can recognize, but they won't mount an immune response to. So some things are better than others when it comes to activating our immune system.
Antigens are macromolecules, usually of molecular weight greater than 10,000, such as proteins and polysaccharides. They are recognized by the immune system as foreign. Individual antibodies are not made against the entire antigen molecule, but rather to particular chemical groups on the molecules known as antigenic determinants or epitopes. Many different antibodies can be made against a single antigen, each antibody reacting with a different epitope. Complex structures, such as the surfaces of bacterial cells, may have many different epitopes. Each different antibody binds only to the correct epitope.
So for as small as a bacterium is or a virus, the immune system recognizes just tiny little pieces of it. And each one of those pieces is called an antigenic determinant or an epitope. Being large in size, being complex in shape and structure is best at activating our immune response. Something that's repeating, or like just think sugar molecules, you know, we have sugars, so it's not complex enough to mount an immune response. So large things, so greater than 10,000 molecular weight units, and then complex in shape, usually proteins make the best, but there are other things. You can throw some sugars on the protein, so glycoproteins, glycolipids, polysaccharides. Those are all possible if they're large enough and complex enough. We've worked with antibodies when we did blood typing, and we'll do that again in lab. And remember, antigens were the things on the surface of the red blood cells, so things the immune system can recognize. And we were concerned about giving a patient blood that was foreign to them because their immune system would recognize it and mount an immune response. Antibodies because I realize antigen and antibody kind of sound the same. You gotta make it to the end of the word. But antibodies has a B in it, and antibodies are the soluble proteins that are in the blood plasma. What we're going to find out today, they're also in your other secretions like tears and saliva and mucus as well, but you kind of have to keep those antigens and antibodies straight. Okay, so antigens stimulate an immune response. The immune response to an antigen is to make antibodies through humoral immunity and B lymphocytes. So the T lymphocytes and the B lymphocytes, they each will have receptors that recognize one thing. So just one particular region of a larger microbe. And so each one is specific, so think targeted assassin. Okay. So those are antigens. So when it comes to particularly the T cells, they read our barcode. So each one of your cells has been tagged as self by a major histocompatibility complex. So major because it's a big deal. Histo, so histology is the study of tissues. And compatible, this is what needs to be matched if someone's going to donate tissues or organs to someone else. Okay, you got your unique tag, or cell surface receptors genetically. Unless you have an identical twin, yours is completely unique from anyone else's. And so this is how your immune system recognizes your cells. Another word for it is human leukocyte antigen, because it's the white blood cells that are going to read this barcode. Like I said, they don't have eyes, so they have to feel. And so these are going to be surface proteins, and they'll have a particular shape. And like I said, you got your shape genetically. So they're shaped like this, and you notice there's a little cleft right here. In this cleft, what a cell is going to do is it is going to sort of display either self-antigens, meaning that cell's doing okay, it's not infected, or if it's infected with something, then it can display things from the virus particle, or if it becomes tumorigenic, it's going to start doing some crazy stuff, and it's going to place things that are not self out here in that cleft. So if a cell is happy, it's going to be a normal self protein on that surface in context of that barcode. If it is infected, then it's going to be something that the immune system is going to recognize this is not normal, this is not right. And we talked about natural killer cells, and natural killer cells, they too they're looking for that barcode. And if it's downregulated, they don't find it. That's one of the things that will trigger their natural killing. Like, well, you're not displaying the right barcode, and it doesn't ask questions because it's natural killer. All right, you see that there's two things. There's two different types of major histocompatibility complexes. One is called MHC class 1, and one is called MHC class 2. So here you go. Everybody has one. Just remember that. All your cells have an MHC class 1. Except red blood cells, which is why all we have to do is type ABO, Rh positive, Rh negative blood groups, and we can give blood to one another. I can't give you a kidney, right? We have to be matched for that. All right, so everybody has one. The cytotoxic T cells, we're going to watch a video in a second, will crawl all over our cells looking for cells displaying a SAD protein, so displaying whatever they're designed to fight. And so if they don't find it, they just leave that cell alone, and that's fine. If they do find their antigen, that's going to mediate cell-to-cell contact and killing. Okay, and so just that's going to be cell-mediated immunity. So that's how they know. This other barcode over here is MHC class 2. This is only going to be on specialized cells called antigen-presenting cells, so macrophages, dendritics, and B cells, and we're going to watch it in a second. This is the one that is going to be read by our T helper cells, and we get activation. Okay, so everybody has one, MHC class 1, and then antigen-presenting cells have a special barcode because it's going to be a different message, and they have MHC class 2. So macrophages, dendritic cells have B cells, so they have class 2, but they also have one because everybody has one, so they get 2. All right. So here's an example. So our cytotoxic T cells, like I said, their job is to kill our corrupt cells, so our cells that have become infected. And so they tell that a cell has become infected by crawling all over our cells, and if they recognize their antigen, then they know that cell needs to go. It's corrupt. We don't want to have it in the body, and so that's what it's going to kill by cell-to-cell contact. So the message of an antigen displayed in MHC class 1 is, yep, I've got our barcode, I belong to cells, but I've become invaded, or I'm cancerous, kill me. And so that's how our cells talk. That's how those cells know, yep, you're not right anymore. We want to eliminate you from the body. And so that the CD8 cells, cytotoxic T cells, they are going to recognize antigens in the context of MHC class 1. An antigen presenting cell, I said they've got MHC class 1, but they also have this special presenter barcode. The reason is they too are going to present things to the T lymphocyte, but the message is different. And I showed you this picture, and I asked if people watch Games of Thrones, and hardly anyone watches Game of Thrones anymore. But when macrophages, dendritic cells eat things, they digest them, because we've learned all about phagocytosis, and then they just display little pieces, parts of them, so those antigenic determinants on their surface. And they go looking for the lymphocyte. Hey, is this on your wanted poster? Is this the thing you're designed to fight? And so it will present that antigen in context of MHC class 2, and the message is, hey, yeah, I'm one of us. I've captured a foreign invader, and this is what it looks like. Help me mount an immune response against it. So they're different messages, which is why there's just 2 different types of markers. All right, let's watch it.
Macrophages and dendritic cells are major antigen-presenting cells. After digesting an antigen, they process parts of the antigen, attach them to MHC2 proteins. These are different from the MHC1 proteins, which most body cells possess. MHC2 proteins identify the macrophage or dendritic cell as an immune cell, so that if it presents an antigen, it won't be destroyed. These MHC proteins then migrate to the cell surface, and thus wear the antigen on the surface of the cell. This presenting of the antigen allows antigen-presenting cells to activate the T lymphocytes in the adaptive immune response. Some dendritic cells are able to enter lymphatic vessels after processing an antigen, and then exit in lymph nodes where they can present to T cells there. So it is this return from peripheral tissues to lymphatic centers which allows these dendritic cells to play a special role in the maturation of T cells based on experience of antigens in the peripheral tissues.
Okay, so dendritic cells are specialized antigen-presenting cells. They're called dendritic cells because they have all these little extensions like dendrites from a neuron. That's where they got their name. Macrophages can do this. B cells can also do this. They have this special MHC class 2, and they can present that to the T lymphocytes. So macrophages, dendritic cells, they are going to be our link between our innate immune system and our adaptive immune system. So we said that innate immunity, one of their jobs is to activate the adaptive branch.